Hi guys, this is a continuation of the DNA chapter. This is sort of part 2 and more concentrated on chromosomes and chromatin. So I'm telling you here that chromatin helps pack the DNA. So I showed you pictures like this of the DNA, and think of it, I have a spool of thread here, and DNA is in this long piece that you can't see there, but if I were to cut this, and I have this long thread here of the DNA in your body, and then all of a sudden I actually scrunched it all up into a ball here that you would now be able to see. So that's what we're seeing when we see these chromosomes. But it wouldn't make any sense to just wrap it up in any old messy way like I did with my thread here. Instead, it coils in a very elegant manner. So on the slide I'm telling you, eukaryotic chromosomes consist of chromatin. So chromatin is a word, a collective term for DNA and all the associated proteins that come with it. What's that mean? Well, in this diagram down here, you see these little balls. They're really a representation of proteins. So if we were to get some proteins and then wrap my thread nicely around these proteins, I wouldn't have sort of a bird's nest like this. Instead, I'd have this organized chromosome. So if I'm telling you here that all DNA in one human cell would form a 2-meter thread. So there's my thread that's going 2 meters long, right? Pretty long thread. And putting it in a form that we can actually organize it in a cell is what's essential. Remember that a cell is going to divide. And you have one cell that's going to divide into 2, and you're going to have to put half of those chromosomes in one and half in the other. And there's got to be an elegant way to do that. So looking at the terminology here a little more closely, these little balls, they're not really a little ball, but they are a protein, and they are called histones. Histones. So if we were to get 8 of those histones together and make an even larger ball here, that is going to be called a nucleosome. So a nucleosome is a group of these histones. Now you can wrap your DNA around these nucleosomes and make it much tidier within a cell. And for us, 23 pairs of these are going to be in your cell. Telling you here on the slide, nucleosomes cluster into bunched up chromatin. So there's the word again, chromatin, before cell division. So before the cell divides, it's got to get organized with its DNA. It's going to wrap around like it's showing you here in the picture, and it's going to end up forming that chromosome made up of genetic material that's called chromatin. So again, I'm telling you, bunched up chromatin, bunched up chromatin is what forms the chromosomes. A highly folded DNA takes up less space and it's easier to move around the cell. So not this long, much shorter. It's easier to move it from one place to another in a cell while it divides, and it's going to be organized. And in the diagram here, it's showing it in the nucleus of the cell, because that's where this is happening. Wrap, wrap, wrap in an organized form, and you end up with a chromosome. This is a table from your text, a mini glossary of chromosome terms, and you'll want to look at this in your book. So it's going to describe what chromatin is, what a chromosome is, what a chromatid is. That's really confusing. Chromatin and chromatid, and a centromere. So I'll let you see those, look them up. You can read it on there here if you pause the video. Continuing on, we know that in order to, if a cell is going to replicate, it needs to replicate its DNA. It's going to double the amount of DNA and then put half in one cell and half in the other cell. So it's multiple steps, and it's quite elaborate, and in a cell biology class you would learn a lot more about it, but for here we're going to summarize that, and I'm going to just show you the very simple steps here. In step 1, you have the strands here are going to unwind. So what used to be the ladder is going to have to open up, and Step 2 then would be each strand is a template that attracts and binds complementary nucleotides, A with T and G with C. So this is the map, right? So if we open it up and then copy, oh yeah, G I need a C, A I need a T, C I need a G, T I need an A. Now I know what that sequence is that needs to be copied. In step 3, each double-stranded DNA molecule consists of one parental, one that started out in the beginning, parental, and one daughter strand, the one that you put new nucleotides onto. And therefore, we can call this semi-conservative replication. Semi-conservative because still half of the parent exists in the new one, so it's not entirely new. For this to happen, I made it sound so simple, but really you would need a lot of enzymes. Every time you talk about an enzyme, the word is going to end in ASES, A's. And so I want to tell you just 3 of those enzymes that are involved in this process. One of them is you need an enzyme to unzip that DNA. So I just told you strands unwind and separate. Well, you need an enzyme to do that. You need to undo the helix. So we're going to call it helicase. Helicase. It's kind of the unzipper, right? The next one is going to have to find the complementary strand. So if A is left unattended here, it's going to need a T. If C is left, it's going to need a G. So in order to find that complementary nucleotide there, you're going to need a polymerase. Polymerase is the one that finds it and checks. It proofreads to make sure it doesn't make an error. Pretty amazing how it self-checks itself. And the last one is the ligase. To ligate something means to glue. So it's the one that's going to bond in the step 3 here, bond these back together again, and now I have one DNA strand and a second after the replication. Okay, so all I need you to know is these 3 steps and these 3 enzymes. That's what you need to know for this replication. It turns out that every 3 nucleotides code for one amino acid. Remember that if you're going to make a long protein, it's going to be made up of component amino acids. And this diagram is showing that. So, for example, AU, which is in messenger RNA, this is the T. So AUG, every 3 of these is coding for one amino acid. The next 3, AUC, codes for another amino acid. The third one, so on and so forth. And here it's linking them together like a necklace and making a polypeptide or a protein. So the DNA then is the code. And every 3 says, okay, those 3, I know it's this amino acid, and grabs it. The next 3, oh yeah, this amino acid, and grabs it. And forms those proteins. Grabs it from where? From the cell. And how did it get to the cell? How did the amino acid get to the cell? You ate it. You ate it in your food. And that's how you're going to get those amino acids. Okay, so let's talk about mutations. What if that replication happens with an error? It could happen, right? It proofreads itself. And it's very efficient, but a small percentage of times is an error. So we can look at it in terms of an analogy. Wild type is what happens normally. So let's look here. Let's say we had a sequence. Instead of telling you C, G, A, C, T, G, C, I'm going to write it out in words. And those words are the one big fly had one red eye. And it's in groups of every 3, right? We're pretending 3 nucleotides at a time. What if you had something like a substitution, where when you go copy that 3rd one, there's an error, and it's a mistake, so it's like error, one big fly had one red eye. That could happen. What else could happen? You could have a frame shift, where you insert an extra one by mistake, and then what would happen? Everything downstream from that one gets messed up. So the one something, you added a Q, and then the big fly had, you notice how everything is wrong here. So this would be a big error, just because of the insertion of one extra nucleotide. What about the deletion? This can happen too. You could have a deletion of 3 nucleotides. So here, the one big, we missed the fly, had 1 red eye. Or an insertion. So you can either delete one or insert one. The one big wet fly had 1 red eye, for example. These are examples of what can happen when we're copying DNA and that replication isn't perfect. And that results in a mutation. Is a mutation terrible? Might be. Is a mutation good? It could be. Or a mutation could have absolutely no effect at all because it was such a small change that it didn't really matter. A big change could cause you not to have an enzyme, for example, and not be able to digest food, which would be a pretty dramatic one. So what you have to remember is that these mutations are absolutely random. They could happen, and the effect could be good, bad, or indifferent. And that's where we're going to stop on this mini video. Thanks.